Well, good afternoon, everyone, and thank you for joining us at a Cancer Center Grand Rounds. This is the annual Kingsbury Lectureship in honor of breast cancer. I have the profound privilege today to introduce Dr. Lisa Newman. She is a surgical oncologist and someone that I have been in awe of for much of my career. She has a clinical and research practice dedicated to breast cancer management. Her formal title is Chief of the Section of Breast Surgery at New York Presbyterian Weill Cornell Medical Center. And she leads a multidisciplinary breast program at the David H. Koch Center, also at New York Presbyterian. Dr. Newman is the new and founding medical director for the International Center for the Study of Breast Cancer Subtypes. And this was recently headquartered at Weill Cornell as part of Dr. Newman's recruitment. She obtained her undergraduate education at Harvard University and attended medical school, completing and general surgery residency at the State University of New York Downtown Medical Center in Brooklyn. She went on to pursue fellowship training in surgical oncology at the MD Anderson Cancer Center and joined the faculty there as an assistant professor before going back to Michigan. Dr. Newman has really been a trailblazer in both her research and clinical care. This is focused on ethnicity-related variation in breast cancer risk and outcome, and the evaluation and management of high-risk patients, including applications for neoadjuvant chemotherapy. She has a very robust research program in disparities in breast cancer risk and outcomes, and has really been lifted up as a national leader in this space. I think on a personal note, she is known as a generous and kind and gifted mentor and clinician. She shared a story with me earlier today about her willingness to give time in the ICU during COVID to call families to update them about their loved ones. And I think this anecdote is a testament to her character. So we are very excited to hear from Dr. Newman today about oncologic anthropology, breast cancer disparities, triple negative breast cancer, and African ancestry. Welcome.
It's a huge, huge honor to be talking to all of you at such an incredible, esteemed academic powerhouse as the Yale Cancer Center. And I first have to express my very deep appreciation to Eric, who, because you have such an incredible leader of your cancer center, somebody who is a force of nature in and of himself, and has been a leader in the breast oncology world for so many reasons and across so many different types of research. But because of his deep dedication to health equity and disparity, simply because of who he is and his nature, it's been a large part credit to him that disparities research is a field of study in and of itself. And so young minds like Rachel and Laios have been able to do wonderful, very, very exciting work in this area because it was made possible by Eric. So I really appreciate how you've stood by all of us over time and made this field of research possible. So I am, over the next few minutes, going to talk to you about my work in what my team calls oncologic anthropology, which is basically the intersection of research trying to understand how African ancestry in and of itself predisposes individuals to some of the high-risk, biologically more aggressive cancers, such as triple-negative breast cancer. Now, the World Health Organization defines social determinants of health as the conditions in which people are born, grow, live, work, age, and a broader set of forces that shape daily life conditions. And nobody would dispute the fact that poverty is clearly going to be a determinant of a poorer health. Since the unequal distribution of wealth in this country leaves communities of color, such as African Americans and the Hispanic Latinx community, with higher rates of poverty. It's not surprising that these issues will go hand in hand with other metrics of socioeconomic disadvantage, such as being uninsured and being unemployed. And then these socioeconomic disadvantages have a downstream effect on health. And so communities of color, such as African Americans and the Latinx community, also have higher prevalence of metrics of poorer health, including being obese and poorly controlled hypertension. And then most recently, we saw how this played out in terms of the consequences of COVID severity. However, the very unique history of individuals with African ancestry in this country has led to a very stark and quite large magnitude disparity in health outcomes for African Americans compared to white Americans. And of course, this dates back to the era of slavery when African ancestry individuals had no autonomy over their own healthcare or the healthcare of their families. But even though slavery was abolished more than 150 years ago, the consequences, the legacy of it stays with us. Over several decades that followed the abolition of slavery, discriminatory banking practices such as redlining permitted the banking industry to essentially leave many generations of African-American families trapped in neighborhoods where they could not own their own businesses or their own homes. And this led to them living over many generations in communities featuring more impoverished school systems, which ends up leaving them with fewer professional and educational prospects. And today we see these communities continuing to be characterized by other features that affect health, such as food deserts, less available healthcare resources. And the mere cumulative experiences over a lifetime of discrimination and racism is now being studied in the field of research called allostatic load, where we are learning that these types of experiences not only have an adverse impact on health, but they also seem to impact on inherent cancer burden. When you look at the specific problem of breast cancer, it's not surprising that these socioeconomic disadvantages that are so highly prevalent in the African American community is going to have an impact on the higher breast cancer mortality rates that we see in African Americans, played out because of the advanced stage distribution that we see for breast cancer related to impaired access to a breast cancer diagnosis and proper treatment. So this very close correlation between socioeconomic disadvantage and African-American identity leads many people to question whether racial ethnic identity has any biologic relevance at all, or is it purely and simply a sociopolitical construct? Well, the answer, of course, is that both defining components of racial identity are present and they are not mutually exclusive. How we self-identify as well as how society labels us will very definitely impact on how we live, on where we live, and on how we access healthcare. However, there are also features of racial identity that are very closely linked to our ancestral heritage. And these ancestral genetic factors will also have an impact on health metrics and on cancer burden. And we've actually known for many decades now that when it comes to a diagnosis of breast cancer, there are indeed other factors aside from socioeconomics that are impacting on the outcome disparities that we see. This slide is now nearly 20 years old and comes from a study that I and some colleagues from the Harvard School of Public Health conducted, where we simply pulled together all of the data in the published literature, looking at breast cancer survival rates for Black women compared to white women, after accounting for some measure of socioeconomics. And as you see here from the forest plot, the African-American breast cancer patients have a statistically significant, nearly 30% higher mortality hazard. And again, this is after accounting for socioeconomic status. Furthermore, there are several features of the breast cancer burden in the African-American community that can't easily be ascribed to socioeconomics. For example, the younger age distribution. And depending on which study you read, up to 40% of African-American breast cancer patients are diagnosed younger than the age of 50, compared to only about 1/5 of white American breast cancer patients being diagnosed in this premenopausal age range. African-Americans, as we will be discussing in this presentation, also have a higher risk of the biologically aggressive patterns of breast tumors, the high-grade tumors, the hormone receptor negative, and the triple negative tumors. We have a higher population-based incidence of primary inflammatory breast cancer. And there's also the very poorly understood higher population-based incidence of male breast cancer in the African American community. And so we definitely need to be exploring tumor biology and genetics if we're going to try to comprehensively understand disparities in breast cancer outcome. Now, you might assume that looking at clinical trials data would be the near perfect way of disentangling racial ethnic identity and socioeconomics from cancer outcomes. And this was the background, the motivation for a really important study that was conducted several years ago by Kathy Albane and colleagues, looking at data from the Southwest Oncology group. And in this study, they pulled together the outcomes for a whole variety of adjuvant therapy trials for different types of cancers, and they wanted to see if equal outcomes were achieved in the context of delivering equal care through participation in a clinical trial. Now, happily, they did show that for the cancers that they looked at, outcomes did equalize given equal treatments, regardless of racial ethnic identity, except when it came to specific cancers. And for the hormonally driven cancers, such as breast cancer and prostate cancer, the African American clinical trial participants continued to have statistically significant worse outcomes. So many of us that have dedicated our careers to disparities research were really excited about Kathy's study because we felt that it was going to usher in a whole new generation of young people interested in studying tumor biology and genetics and looking at cancer outcomes. We were therefore a little dismayed at the fact that Time.com reviewed her study and called it an example of racial profiling in medical research. So we were happy that her study was getting a lot of publicity, of course, but this title was a little dismaying because, of course, racial profiling has a lot of very negative connotations, and appropriately so, when it comes to racial profiling in, for example, the criminal justice system. But when it comes to cancer biology and studying cancer outcomes, racial profiling, if you will, is really just an example of epidemiology. And we absolutely have an obligation to study all of the characteristics of our patients when we're trying to understand why some cancer patients have a better or a worse outcome than others. And this does include characterizing the racial ethnic identity of our patients. An example of how important this racial characterization of our cancer patients is shown on the graphic to the left on this slide, where we're looking at the most basic of epidemiologic statistics, population-based incidence rates of breast cancer and population-based mortality rates from breast cancer over the last several decades, as documented by the Surveillance Epidemiology and End Results Program. And what we see here is that over time, incidence rates of breast cancer historically have been lower for Black women compared to white women, but the rates typically changed in parallel, indicating comparable effects of different environmental factors. But for mortality rates shown by the two curves at the bottom of this slide, the mortality rates from breast cancer were equal for Black women and white women until we reached the early 1980s. And at that point, the mortality curves separate predominantly because of declining death rates in white women, but largely unchanging rates in Black women. And this is probably because the advent of tamoxifen as our first endocrine targeted therapy for breast cancer, the effects of tamoxifen become apparent by the early 1980s. But as shown by the bar graph to the right of this slide, since African American women have significantly lower frequencies of the estrogen receptor positive cancers and higher rates of estrogen receptor negative tumors, we are just not benefiting from the advantages of terrific systemic therapies such as tamoxifen to the same degree as our sisters from other racial ethnic backgrounds. So we basically, by the early 1980s, are seeing the unmasking of differences in tumor biology between African American and white American breast cancer patients. So now, of course, we've gone far beyond simply characterizing breast cancer as the dichotomous hormone receptor positive versus hormone receptor negative tumors. And we know that breast cancer is comprised of an entire spectrum of intrinsic tumor subtypes, with the basal subtype being one of the more virulent subtypes. The patients that we see every day in clinic are not necessarily going to get complete genomic profiling done on their tumors. So we use immunohistochemistry to look at estrogen receptor, progesterone receptor, and HER2/neu expression as a convenient way to have a surrogate for identifying the most aggressive of these tumors. And the triple negative breast cancers do tend to correlate with identifying a patient that has an intrinsic basal type tumor. It's not a perfect correlation, but it is pretty close. And as shown by the curves on the top right of this slide, women that have the triple negative breast cancers at every stage of breast cancer diagnosis have worse outcomes compared to the women who have the non-triple negative breast cancers. And we now know from many studies that have been done that African-American women have higher frequencies of triple negative breast cancers, regardless of the age at which they're diagnosed. And we have higher frequencies of triple negative breast cancer, regardless of the stage that the breast cancer is diagnosed. Now, there's been an interesting phenomenon over the past couple of decades where the population-based incidence rates of breast cancer have been rising disproportionately in African-American women. And now, over the last 10 years or so, breast cancer incidence rates are pretty much equal for Black women and white women. But those rising incidence rates of breast cancer in Black women, coupled with our higher incidence of the triple negative breast cancer, has resulted in a widening of the mortality gap. And today we see about 40% higher breast cancer mortality rates in the African American community compared to the white American community. And it's impossible to have a discussion about triple negative breast cancer and disparities without making some comment regarding mammography screening recommendations. And as I'm sure all of you are aware, the United States Preventive Services Task Force has been advocating pretty aggressively for average risk American women to delay initiation of screening mammography until they reach age 50. Many of us that are dedicating our careers to studying breast cancer disparities are really concerned about this broad recommendation because waiting until age 50 for mammography screening can result in an even worsening of the delays in diagnosing biologically aggressive tumors such as triple negative breast cancers in African American women who are already at higher risk for getting these aggressive tumors at younger ages. And so this screening recommendation will likely worsen the disparities that already exist. Now, the critics of screening mammography are always quick to point out that mammography is not going to be the be-all, end-all answer to addressing disparities, because it is true that triple negative breast cancers are more challenging to detect on screening mammography, and they're more likely to present as the palpable interval cancers in women that are getting their screening mammograms every year. However, we do have very strong data showing that early detection of triple negative breast cancer does still make a difference. And an example of those data are shown in the two tables on this slide, where investigators from Memorial Sloan Kettering and from the National Comprehensive Cancer Network have both demonstrated that triple negative breast cancer, when it's diagnosed at a small size, no larger than 1 centimeter in size, and with nodes negative, and these are by and large going to be screen detected triple negative breast cancers, these tumors have very good outcomes regardless of whether the patients receive adjuvant chemotherapy or not. Now, those two studies that I showed on the previous slide were looking at early detection of triple negative breast cancer, but they weren't necessarily looking at mammography screen detected triple negative breast cancer. And so to address the question of how effective screening mammography is in outcomes, in improving outcomes from triple negative breast cancer, our group pulled together the data on triple negative breast cancer patients from the metropolitan Detroit area and the Henry Ford healthcare system and the Weill Cornell New York Presbyterian Hospital network triple negative breast cancer patients. And we looked specifically at outcomes for these patients with triple negative tumors based upon whether it was screen detected disease or not. And we looked at a whole bunch of different factors that might also impact on outcomes from triple negative breast cancer. For both the white and the African American triple negative breast cancer, having a mammography screen detected tumor was the strongest predictor of a patient that was going to have a good outcome. So we do indeed have data that mammography screening is effective at early detection of triple negative breast cancer, and it does yield some benefits in terms of improving outcomes. And that benefit was actually strongest for the African American women. We've also been looking at whether or not there might be some precursor lesions in benign breast tissue, identifying women that are at higher risk for getting a triple negative breast cancer, and whether or not the benign breast patients who require biopsies will still have a higher rate of triple negative breast cancer correlating with racial ethnic identity. As all of you are aware, the number of benign breast biopsies that a patient has does correlate with a higher risk of a future breast cancer, probably because it's identifying hyperproliferative changes in the breast. And this is why number of biopsies is integrated into many of our risk prediction tools, such as the Yale model. But by and large, multiple biopsies is a predictor of having an estrogen receptor positive breast cancer. So we utilize the Henry Ford Health System benign breast disease cohort to look at whether fibrocystic breast biopsies predicted for higher rates of triple negative versus hormone receptor positive disease in our Black compared to white patients. And we had a very large cohort of more than 6,000 women who had had benign breast biopsies with robust follow-up of more than 10 years. And in as evidence that these women were receiving equitable treatment over the years, we actually saw comparable rates of subsequent breast cancers in these women with fibrocystic breast changes, regardless of whether they were Black or white. And we saw comparable stage distribution for the cancers that did develop in these patients. However, as shown by this curve graphic at the bottom of this slide, the African American women with benign breast biopsies had about a 4-fold higher risk of getting a triple negative breast cancer compared to the white American fibrocystic change patients. And so there does, at least from our experience, seem to be something inherently different about the mammary tissue of African American women, increasing the susceptibility for these biologically aggressive tumors. Another interesting question to ask is whether or not outcome disparities will persist after you stratify for tumor phenotype. And I'm not going to belabor the data on this very busy slide, but suffice it to say, there are actually a number of studies suggesting that when you adjust for stage and treatment, that the outcomes from triple negative breast cancer patients might actually be fairly comparable for Black women and white women. However, there are numerous studies showing that for hormone receptor positive breast cancer, the disparities persist. Now, whether or not these disparities in outcome in hormone receptor positive disease are related to differences in tumor biology or difference in response to endocrine treatment or just variation in compliance with the several years that we recommend for endocrine therapies, these are all questions that continue to be under study. But now we are starting to generate some answers to those questions because brilliant researchers who've been conducting terrific studies of gene expression profiling in women with hormone receptor positive, HER2 negative breast cancers are now starting to look at their data based upon stratification for race ethnicity. The TAILORx investigators have recently shown that for women undergoing Oncotype 21 gene recurrence score testing for hormone receptor positive, HER2 negative, and node negative breast cancers, that in women with the intermediate scores, the African American women have notably higher rates of recurrence and mortality, even after adjusting for these intermediate range scores. And then very recently, the investigators for the responder trial reported in the San Antonio Breast Cancer Symposium that among women looking at these 21 gene recurrence scores and whether or not they predict for benefit from chemotherapy in the setting of women with node positive disease, they similarly showed that the outcomes for the African American patients were significantly worse compared to the outcomes for the white American patients. And again, this is after stratifying for the 21 gene expression score. Many investigators have been looking at data from the Cancer Genome Atlas to try to take a deeper dive, basically, into looking at tumor biology between Black women and white women with breast cancer. And I'm summarizing just a few of the studies that have been published utilizing TCGA data on this table. But all of these studies are basically looking at the same group of, more than 700 white American breast cancer patients and about 170 African American patients that have contributed tumor tissue and clinical information. So it's not surprising that all of these investigators have identified similar patterns. PAM50 subtyping definitively showing that the African American women not only have higher frequencies of the triple negative immunohistochemically defined phenotype, but we also have higher rates of the intrinsic basal subtype. The African American patients are more likely to have TP53 mutations and fewer PIK3CA mutations, which goes along with the higher frequency of triple negative and lower frequency of hormone receptor positive tumors in these patients. And the phenomenon of seeing higher rates of these biologically aggressive estrogen receptor negative and triple negative breast cancers in women with African ancestry is actually not something that's unique to the United States. And other countries, the UK, Switzerland, Brazil, investigators from these countries have also published data showing that their African ancestry breast cancer patients are more likely to have estrogen receptor negative and triple negative breast cancers compared to their non-African ancestry breast cancer patients. So this is why our group has been very excited about looking at international data, and in particular, looking at the breast cancer burden of women on the continent of Africa to try to tease out the answer to the question of whether or not African ancestry in and of itself is associated with some heritable marker predisposing to risk for triple negative breast cancers. And this, I think, opens the door not only to exciting ways to understand disparities, but also very novel ways of trying to understand the pathogenesis for triple negative breast tumors. So this is just a snapshot of some of our most basic findings, looking at the frequency of triple negative breast cancer in women from Ghana, representing Western Sub-Saharan Africa, compared to the frequency of triple negative breast cancers in women from Addis Ababa, Ethiopia, representing East Africa. And we see quite high frequencies of triple negative breast cancers in the Ghanaian women. About half of them are triple negative. But the frequency of triple negative breast cancers in the Ethiopian women is very low at about 15%, similar to what we see in white American and European patients with breast cancer. The frequency of triple negative breast tumors is intermediate for African American women between the rates that we see in Ghanaian women and what we see in white American women. Now, the American Cancer Society, brilliant investigator Ahmadine Jamal, has published data that are comparable to what we're seeing in our international data set. Dr. Jamal has published data looking at the frequency of ER-negative breast tumors, which of course are a subset of the triple-negative breast tumors, in white American breast cancer patients, African American breast cancer patients, and women born in West Africa but whose cancers were diagnosed and treated in the United States, and women born in East Africa but whose breast cancers were diagnosed and treated in the United States. And similar to our international data, Ahmadine found that we see the highest frequencies of ER-negative tumors in the African American and West African born patients, and the lowest frequencies of ER-negative tumors in the white American and East African born patients. So this is where we've coined the nomenclature of oncologic anthropology to try to explain these patterns. And of course, as we all recall from grade school social studies, the transatlantic slave trade brought the ancestors of contemporary Western Sub-Saharan Africans across the ocean to serve as slaves in the colonies. And so today, as contemporary African Americans, we have quite a bit of shared genetic ancestry with the contemporary Ghanaians representing Western Sub-Saharan Africans. But the slave trade from East Africa largely brought the ancestors of contemporary East Africans and Ethiopians further eastward to the Mideast and to Asia. And so as African Americans, we don't have quite so much shared ancestry with Ethiopians. And so if there is something of a heritable nature related to African ancestry predisposing to triple negative breast cancer, it's likely something specifically related to Western Sub-Saharan to African genetic ancestry. So one of our terrific and brilliant research partners for my research team, the International Center for the Study of Breast Cancer Subtypes, is Dr. John Carpton, who runs the Translational Cancer Research Program out at USC. And we've been really trying to work quite hard to get more of our colleagues in the oncology research world to look at the genetics of race and ethnicity and to quantify germline ancestral genetics with cancer outcomes as a way of trying to understand disparities better. So about 5 years ago, I was absolutely thrilled to be able to recruit one of John's mentees, Melissa Davis, who is a card-carrying PhD geneticist, to serve as the scientific director for our International Center for the Study of Breast Cancer Subtypes. And our international team for the last nearly 20 years now has been building up this biobank, a biorepository of tumor specimens for somatic tumor tissue studies and saliva specimens, as well as blood specimens suitable for germline genetic studies from different parts of Africa. And so it was really exciting to get Melissa to serve as our basic science research leader so that she could use her tools to tease out some of these differences in understanding the genetics of African ancestry. And for most of Melissa's career, she's been a world-leading expert in studying a particular gene called the Duffy gene, or the Duffy antigen receptor for chemokines. And there's a particular variant of the Duffy gene that is seen almost exclusively in individuals that have Western sub-Saharan African ancestry. It's widely called the Duffy null gene variant, and therefore this Duffy null variant is an ancestry informative marker, informative of African ancestry. Now, Melissa's been studying Duffy null for most of her career. Other investigators have kind of happened along Duffy null in the context of other studies looking at disparities. And this slide is summarizing the data from a study that came out of the AMBER Consortium, a collaborative group of investigators looking at breast cancer disparities related to race. And in this particular publication, the AMBER Consortium investigators were looking at levels of different circulating chemokines that might be associated with cancer and in particular breast cancer risk. And they wanted to see if the levels of these different cytokines were different between Black women and white women who had not yet been diagnosed with breast cancer. And they did identify a handful of cytokines that differed between the Black women and white women. And then when they did genetic analyses, they found that these differences were explained by the presence of the Duffy null genotype. We've also learned over time that the Duffy null variant is the variant that's responsible for a phenomenon called benign ethnic neutropenia, which is the fact that African Americans tend to have a lower circulating white blood cell count, which doesn't have any biologic significance, but it is a numeric pattern that's seen pretty consistently. And some investigators are now looking at whether or not Duffy null may be implicated in transplant rejection disparities. And we're obviously looking at it in breast cancer. Others are looking at it in prostate cancer disparities. Unfortunately, however, when you look at the literature globally, there is a huge gap in terms of what we know about how African ancestral genetics impact on cancer risk, because so few of the genomic studies have included significant numbers of individuals with African ancestry. And as shown by this study from Cell, only about 2% of individuals contributing to genome-wide association studies have had African ancestry. So we were really excited to have Melissa work her magic with her genetics skills to apply them to our international biorepository, which again has been amassing specimens for nearly 20 years. So Melissa did a really cool study where she looked at Duffy null compared to a series of other genetic variants that have been potentially linked to risk of breast cancer and hormone receptor negative, triple negative breast cancer. And in working with this other brilliant researcher, our geneticist, biostatistician, Yale Chen from the Henry Ford Health System, Yale was able to show that the presence of this Duffy null variant was by far and away the strongest determinant of having a triple negative breast cancer versus having a non-triple negative breast cancer. The phenomenal anthropologist, Dr. Sarah Tishkoff, has shown us very nicely, as demonstrated by this graphic, that many of the ancestry informative markers that we look at aren't markers, variants that developed randomly over time. Many of them actually represent evolutionary selection pressure over our ancestors to allow our ancestors to survive different threats to longevity, threats to life expectancy related to infectious diseases, related to climate, related to food sources. And then today, when we look at the descendants of those populations, you can continue to see many of these ancestry informative markers, regardless of where the descendants reside over the globe. The Duffy null variant is just one more example of such a variant that was acquired over the millennia as a consequence of evolutionary selection pressure. The Duffy null variant is something that became apparent, that was adopted in Western sub-Saharan Africa many thousands of years ago, linked to the need to have some resistance to malaria. And malaria became endemic in Western sub-Saharan Africa because of the tropical nature of the geography there, with the many watery areas and low altitude areas supporting the life cycle of the mosquito, which of course is the host for the malaria parasites. And there are other examples of variants that were acquired to confer some resistance to malaria. The thalassemias seen in European Mediterranean populations, sickle cell. The Duffy null variant doesn't have quite as many of the adult health consequences as those variants do, and therefore the Duffy null variant is seen in nearly 100% of the descendants of Western sub-Saharan Africans. Something occurred about 6,000 years ago called the Bantu expansion, where Western sub-Saharan Africans migrated across the continent to populate various areas of East Africa and South Africa. And while many of those areas have more mountainous areas that do not support the mosquito life cycle, so they have a different history of endemic malaria in those parts of the continent. But with the Bantu expansion, the Duffy null variant did track across the continent of Africa, and you see varying degrees of admixture and the presence of this Duffy null variant in those regions of Africa. Just as with the transatlantic slave trade, the Duffy null variant came across to the Americas, and with the genetic admixture that we see in African Americans, this results in about 2/3 to 3/4 of African Americans expressing that Duffy null variant. And if you overlay a map of the frequency of triple negative breast cancer in different parts of the world with a map of the Duffy null variant, there's actually a pretty close correlation. So we've been exploring ways to understand how to connect the dots between this Duffy null variant and the risk of having a triple negative breast cancer. The reason why that Duffy null variant confers some resistance to malaria is because if you possess the Duffy null variant, you do not express the Duffy protein on your red blood cell, and the Duffy protein on the red blood cell is kind of the entry portal for the malaria parasites to get into the red blood cell and cause the disease. So now what we are learning, and the work that's ongoing in Melissa's lab, is seeking to better understand how this Duffy protein and lack of the Duffy protein on the red blood cell impacts on circulating chemokines, which would explain the findings of the AMBER consortium that I showed you previously, and how this may have a downstream effect on the mammary tissue microenvironment and the inflammatory, the immune landscape of the mammary tissue, which can then have an impact on the types of breast tumors that develop. Melissa has also been doing work with the Cancer Genome Atlas, looking at tumor tissue expression presence of the Duffy protein. And you see, as you would predict, lower levels of Duffy protein in the breast tumors of African American compared to white women contributing specimens to TCGA, and the lower presence of Duffy tends to correlate with worse prognosis across the different phenotypes. So this phenotype agnostic, if you will, effect on tumor tissue, tumor outcome may be what's explaining what we see in looking at the impact of race ethnicity on outcomes in women that have ER positive disease, as we saw from the TAILOR-X and the responder trial. Many people are doing very, very exciting work seeking to subtype the triple negative breast cancers. And a lot of this work was pioneered by the Vanderbilt group, identifying about 6 different intrinsic triple negative subtypes initially. However, the publicly available data sets that contribute to the definition of those different triple negative subtypes largely came from communities that had very few, if any, African ancestry individuals. So we really don't know if those triple negative breast tumor subtypes are applicable to the African ancestry populations that have a higher inherent risk of developing these triple negative subtypes. In working with Clayton Yates, who used to be at Tuskegee and now he's recently relocated to Johns Hopkins, Clayton has also been utilizing data from our international biorepository and has identified the fact that the triple negative breast tumors of African American women does seem to have different signatures compared to what we see in white American women. Also utilizing data from TCGA, Melissa's been looking at the impact of tumor-infiltrating, tumor-associated lymphocytes on breast cancer outcomes. And we typically think of these tumor-infiltrating lymphocytes as a favorable prognostic feature. But these are unpublished data. In these preliminary data from TCGA, the relationship seems to be flipped for African American women, where the higher frequency of tumor-associated lymphocytes seems to be an adverse prognostic feature. Data correlating with these findings from TCGA have been published again by members of the AMBER Consortium. This comes from a study published by Christine Ambrosoni and her group, where they looked at the tumor microenvironment signature of breast cancers from African American and white American women. And while they did show that African American women tended to have a more robust tumor infiltrating lymphocyte content to their tumors, the lymphocytes of the African American women were more likely to have this T-cell exhaustion signature, as they called it, and so their function was different compared to what we see in what they saw in the white American women with breast cancer. And this was an intriguing study that was published in Cell just a few years ago, where some investigators were looking at immune cells that were basically primed with specific pathogens, infectious agents, and then looking at the response of these immune cells from patients that were African American compared to white American, and they saw very distinct and different responses in terms of the immune activity of these immune cells when they're linked to different pathogens. So you can only imagine that if the immune cells of African ancestry individuals are responding differently to infectious diseases compared to the immune cells of white individuals, there could easily be differences in the way these immune cells function in terms of cancer biology. So Melissa has been continuing to utilize our international data set in conducting other studies looking at the triple negative breast cancer risk alleles. And I'm going to go through these next few slides quickly in the interest of time. We've also been working with investigators from the University of Michigan, been creating PDX models based upon our international studies. And then very recently, a couple of months ago, we were really excited about our work with triple negative breast cancer subtyping, which was the cover article for Cancer Discovery a couple of months ago. So to us, that was like being on the cover of Vogue magazine. So we were very, very thrilled about this. And we were able to show that looking at genetic ancestry does have independent meaning compared to self-reported ancestry. And there were several hundred genes linked to genetic African ancestry that you don't see if you look only at self-reported racial ethnic identity. And this is another slide that came from that particular publication where we're just demonstrating the genetic admixture of populations in different parts of the world, specifically looking at Ghanaian patients, African-American patients, Ethiopian patients, and European ancestry, white American patients. And it's an example of how much more you can learn about genetics of disease by drilling down into the genetic ancestry. And African Americans have tremendous genetic admixture compared to either Africans or European ancestry individuals. And you can't rely upon self-reported ancestry. There are 3 individuals in the European ancestry group. These are individuals who self-reported as being white, but they had between 30 and 80% of African genetic ancestry. So you definitely miss information if you rely exclusively upon self-reported racial ethnic identity. And there are other examples of how genetic ancestry might be correlated with health. Apolipoprotein 1 is an African ancestry variant that has been linked to severity of kidney disease. And we all know that end-stage renal disease is more prevalent in the African-American community. This particular variant is actually a variant that was acquired to develop resistance to the African sleeping sickness disease. Also, our wonderful colleague out in California, Laura Feierman, has been doing similar work looking at Latinx individuals. And Laura has demonstrated that extent of genetic Native American ancestry reduces the risk of getting breast cancer. On the other hand, higher extent of European ancestry is associated with a higher risk of getting breast cancer. Other investigators have been trying to figure out the germline genetic ancestral causes of the BRCA founder mutations and have been potentially linking some of those founder mutations to fertility over the millennia. So we've, of course, been very excited about our international group with respect to these different research avenues, but it's also been an incredibly rewarding experience from the perspective of being able to invest in the cancer care resources of the facilities where our partners work. And our mission statement is to reduce the global breast cancer burden through advances in research and delivery of care to diverse populations worldwide. A few examples of how we've been making those investments are shown here. We've been able to establish immunohistochemistry training programs so that our colleagues can perform their own immunohistochemistry on site and actually characterize the cancers of the patients that they're treating. We've established core needle biopsy training programs so that they can make their diagnoses more efficiently. And accelerated through the COVID experience is that we've been able to stay in very close contact utilizing Zoom meetings and telemedicine tumor board discussions. And now that our program is headquartered at Weill Cornell in New York, we are able to align our international studies with the robustly diverse population of New York. And we have our New York-based breast cancer campuses in Manhattan, Brooklyn, and Queens, which has tremendous diversity in those communities. And a lot of our work today is being done in conjunction with the Englander Institute of Precision Medicine. Whenever I talk about breast cancer disparities, I always include these survival rates of 60%, 43%, and 20%, which have absolutely nothing to do with cancer outcomes, but these are the survival rates for the first class, second class, and third class cabin passengers of the Titanic. And even though my own career in breast cancer research and studying disparities has been heavily rooted in trying to understand tumor biology linked to African ancestry, we always have to end just the way we began this discussion, with an expression of the fact that outcome and the ability to survive any threat is going to be related to access to care. And just as the third class cabin passengers of the Titanic did not have equitable access to the lifeboats, it unfortunately and tragically remains true that communities of color, including African Americans, do not have equal access to cancer care, screening, research opportunities. And as stated by Dr. Martin Luther King Jr., of all the forms of inequality, injustice in health is the most shocking and inhumane. We saw this injustice in the COVID experience. And as you guys know, it's been projected that as a consequence of the COVID shutdown and its downstream impact on cancer research and cancer screening and treatment, we're probably going to see an excess of about 10,000 deaths from colorectal and breast cancer in the next 10 years. Because the COVID recession was disproportionately severe in communities of color, we really do have to be proactive in making sure that we protect our disadvantaged communities from experiencing these excess deaths disproportionately. We want to get rid of all of these excess deaths, of course, but unless we support our safety net hospitals, which were disproportionately financially devastated by the costs of COVID care, unless we protect our advocacy and philanthropy groups that provide a lot of our free screening programs, and unless we really work with our hospital leadership to make sure that they don't cut navigation programs, outreach programs when they're trying to balance their budgets in the wake of the COVID experience, we're going to have an exacerbation of these types of mortality gaps. But I am an optimist, and I do know that by working together, we are going to be able to eliminate these disparities. And I look forward to strengthening all of the partnerships that are already ongoing and bringing researchers from different areas together to try to conquer these problems from all different angles. And in closing, I just want to thank all of the wonderful teams that have supported our research over the years. And in closing, I do also want to acknowledge this phenomenal woman, my sister, Deborah Newman, who passed away almost a year ago today from an incredibly aggressive and virulent inflammatory form of triple negative breast cancer. And my sister Debbie, a Princeton graduate, former U.S. prosecutor, she's a perfect example of how socioeconomics are not the exclusive explanation for breast cancer disparities. And so it's in her memory that I and my research team continue the work that we've been doing. So I do thank all of you for your time and attention and for inviting me to deliver this presentation.
Thank you so much, Dr. Newman, for sharing your extraordinary research with our group. I'd be happy to start with any questions from the audience before we turn to the Zoom chat. Go ahead.
One point that I really enjoyed that you underscored was how the self-reporting of race definitely does not usually capture really what the person is. And that really had me thinking about populations might be overlooked in these sort of studies that you are segregating specifically. But with the onset of decreased sequencing costs and ease of access to that sort of data, do you and your groups plan on looking at those populations and identifying specific genetic factors? And if you're not already, have you seen whether specific races have become dominant types compared to others?
Thanks so much for the kind comment. And I totally agree with your points that we definitely have an obligation to look more closely at genetic admixture. And you're right, self-reported race, I mean, it's really primitive. And as cancer researchers, we've been so late to bring the technology of quantification of ancestry into our work. But the general population has been doing this for years. I mean, millions of people are purchasing these products where they spit in a cup and get back their pie diagram of where their ancestors come from. So, I mean, I think that this type of work should be routine in our studies, because we do have the technology and it's so much more precise and meaningful. I agree with you in trying to understand cancer outcomes. Now, we do need to look at self-reported race as well, because self-reported identity does have very, very important relationships to health equity and services that are available to some communities and not available to other communities. But we can't overlook the genetics components. So thank you.
Dr. Weiner.
Okay. So Lisa, thanks for a great talk, and sorry about your sister, and thanks for sharing that. I want to drill down a little bit on ER positive tests.
Mhm.
And of course, adherence to endocrine therapy may be an issue. Not only adherence, but doctors prescribing endocrine therapy, which isn't really adherence, which you think of as a patient issue, but it may be a doctor issue. But the other question, the question I have is, to what extent do we know whether simple things like ER expression vary across race, or whether luminal A versus B percentages vary?
Yeah, it's all terrific questions. Now, from our biorepository, we definitely see higher frequencies of those weakly positive ER tumors, 1 to 9% in the African ancestry patients compared to the white patients. I can't say that I've seen that broadly in publications, however, because we usually just talk about ER positive, ER negative using the ASCO-CAP guidelines. But in our database, we do see that. So I do think that it's a spectrum that's present. I think you're probably right that there are variations in how endocrine therapies are prescribed and how much attention we pay as healthcare providers to adherence to treatment based upon what our patients look like. I think those are very real issues. I am so excited that people like you, the leaders in clinical trials, are paying attention to this in looking at these gene expression profiles. They're going to be, I think, incredibly powerful in studies to come.
Lisa, phenomenal presentation and a body of work that features beautifully molecular epidemiology, clinical questions, and what you highlighted, cultural anthropology. I think you have devoted a lot of time. If I take the same discussion in pancreas cancer and TCGA, I'll tell you there are 5 patients of African American ancestry. And that's the disconnect that I'm always struck that we know so much that biologic heterogeneity matters. And clinicians have always been able to, people like you've been able to answer fundamental questions. Yet my worry is in our passion to, and you brought the Time article, right? And I've seen the same, that in our desire to be equal, we're perhaps missing on those essential things you pointed out. How do we teach that? We're an academic organization, and you've highlighted how clinicians can interact with basic scientists. How do we as leaders make sure we're pointing that out to the next generation?
Yeah, great question, and it's something that we all have to keep working on over time. I am very optimistic. The fact that people are documenting cancer outcomes stratified by racial ethnic identity where it wasn't necessarily documented in the past, the fact that there's a lot of momentum to look at the diversity of our workforce and to develop pipeline programs when very little attention was paid to this in the past. People would remark upon the lack of workforce diversity, but everybody said, well, this is a problem that no group can address overnight, and so nobody tried to do anything to address it. But I think that the COVID experience, horrific as it was, the COVID experience with disparities in COVID outcome hitting us literally in the face, coupled with witnessing the horrific murders of George Floyd, Breonna Taylor, so many others in the hands of law enforcement, all of those events happening together made this an extremely unique moment in time. And so I think that the efforts that we're seeing now in achieving health equity are real. And I think that it's going to make a difference and accelerate the pace of disparities research and accelerate the pace of trying to achieve health equity.
Breast cancer sort of luminaries in the room right now, and where breast goes, other cancers follow. Comment, people have no problems giving their spit saliva for 2 minutes. In fact, in Baltimore, we had a Raven scheme and they wanted to give the patients, the audience, people coming in, free testing. And Antonia Wolf and a bunch of us wrote an editorial saying that was a horrible idea without any guidance. So people are willing to give their data, yet if they come in for an IRB trial, you're gonna make it really hard. And so academia makes it very hard for research to happen. And we put so many barriers. So if you look at clinical trials, it's really hard for people with limited means to come to Cornell or to come to New Haven. So how do we change that? Because who has the best interest in understanding that?
Yeah.
The patients, and yet we make it so complicated. And I'm going to urge all of us to sort of think about how do we break those barriers to make this, because it's fundamental. As you said, this was an evolutionary mechanism
Yeah.
to survive in Africa, right? Malaria is endemic, and now we're seeing like it makes your chemokinin, your inflammatory, you have this response, but you know, it's the flip side. And so I think that there's a piece for us as leaders in the field to say, are we making research just too complex, and simple things like my background probably affects how I respond to the world, because evolutionary and driven into our DNA over thousands of years, right? And we're trying to fix that. So,
Yes. You are so right. Yeah, and you hit the nail on the head, I think, in talking about how we've inadvertently created barriers to diverse populations contributing to research. You know, and many studies show that African American cancer patients are at least, if not more likely, to participate in clinical trials if they're offered the opportunity to do so. And we've created all these barriers where clinicians are less likely to offer them for a whole host of different reasons, implicit biases. Sometimes it's just incidental, well-meaning physicians who are worried that they're going to alienate their patients if they offer an African-American cancer patient a clinical trial for fear that the patient might think that they're being treated like a guinea pig. But we have to get over those types of things, and we have to offer all treatment opportunities to all of our patients. Even when it comes to our IRBs, you know, we have all these regulations that try to protect people against coercion. And so we don't want to offer financial incentives to patients for fear of the more vulnerable patients being coerced. But our socioeconomically disadvantaged patients need that financial support in order to take the time off work so that they can come in for the visits. So it's such a broad sweeping problem to try to meaningfully and thoughtfully get rid of some of these barriers that we've inadvertently created in trying to protect our patients against research injustice. The research abuses, you know, we can't let those come back, but we do also have to be thoughtful and make research easier for our patients.